We're going to be finishing up the cell unit with a couple more lessons. This one is going to actually parlay into the biochemistry unit as well, as we'll be covering not just the last part of cells, which is to really look at the cell membrane, but we're also going to reinforce some more specifics on lipids, some of the specifics that we did not get into in the biochemistry unit. You also would have addressed some of the concepts on lipids that we're going to look at today in your Heart Health Public Service Announcement project. So hopefully some of the things that I'm going to be talking about, hopefully they're not completely new to you. So our main goals for this lesson, our main objectives, are to take a look first of all at the structure of lipids. We're going to evaluate foods that have that are high in the different types of lipids. So we're going to be looking at basically good fats and bad fats. We'll go through that quickly, as again you would have already done that in the PSA. And then we're going to take a look specifically at the cell membrane and the structure of the cell membrane and talk about how it is what we call the fluid mosaic model. So we are going to address all of those topics here today. The overlying objective for today's lesson is to identify the structure of macromolecule, lipids, and the cell membrane through Mr. E-led instruction and online resources. Quick step back to the biochemistry unit, and again you'll need to know this anyway for our upcoming cumulative cell test. We have our 4 groups of macromolecules. Remember, there are specific requirements for macromolecules in terms of their function, and the main idea is that they are involved in making up the structure of living things. Carbohydrates, nucleic acids, proteins, and lipids. Go ahead and get these 4 down, and then take a moment and identify the monomers of each one. Let's try and do this without looking. Just go ahead and write down the monomers of each one. Okay, so we have for carbohydrates, the monomer being a simple sugar. We said that things like glucose, anything that has that 1 to 2 to 1 ratio of carbon to hydrogen to oxygen is going to fit into that category. For nucleic acids, we have a nucleotide. All right, and then for proteins, we have an amino acid. All right, for lipids then, as we've learned, we have a fatty acid, also a glycerol, and today is when we're going to be getting into the specific structure of that. So far we have learned the structure of a carbohydrate, the structure of an amino acid. We will be looking at the specific structure of a nucleotide as we dive into RNA and DNA after the new semester begins. All right, so let's go ahead and start with fatty acids, the building blocks of lipids. As you would have identified in the Heart Health PSA, there are good fats and there are bad fats. And I want you to remember that the purpose of fat is for long-term energy storage. All right. Lipids also have a structural function, which is going to be specific to the cell membrane. All right. So for fatty acids, we've got good fatty acids, we've got bad fatty acids, and that's what we're going to dive into. Let's start on the good side, all right, with this. And by and large, our good fats are going to fall into a unsaturated category. All right, there's going to be good unsaturated fats, and then over on the bad side, there's also going to be a bad version of unsaturated fats. You've probably seen on your food labels something called trans fat, or you might see an advertisement that says this food does not have trans fat. Okay, trans fat is about the worst type of fat that a human can consume because it has been modified. So it's a fat that has been what we call hydrogenated, and we'll get into that here shortly, which makes it even more unhealthy than our regular bad fats, which are going to be saturated fats. All right, so that's where we are at there. For each one of these groups, we're going to talk about some basic food types that fit into these categories. and we're going to talk about chemically how they are composed, chemically how they are made up. So let's start with the good ones over here and the good unsaturated fats. All right, so for our food types here, we're going to see things like avocados, nuts, and salmon. These are good fats because of their chemical makeup, because of how they are made chemically. All right, so that's our next concept, is to look at the chemical makeup of these. All right, so these types of fats are going to have one double bond. Okay, so as you're identifying fatty acids, that's going to be one of the most important parts of unsaturated fats. Okay, if you see a chemical chain and you see at least one double bond, all right, then that means it is going to be some type of unsaturated fatty acid. Now, the number of double bonds is going to determine if it's a monounsaturated fat, all right, or a polyunsaturated fat if it has more. And in the unsaturated fat category, the more double bonds it has, the healthier and better it is for the human body. And we'll get into that here shortly. But first, make sure you get down that chemically there's going to be at least one double bond, and that double bond gives that chain the ability to bend. All right, so the cis monounsaturated fat, like I do with a lot of these big words that we look at in biology, let's go ahead and break down the actual word itself. So the first part here, cis, is going to mean same side. So cis is going to mean something is going to be on the same side, and we'll take a look at what that is here in a moment. And then mono, well, I'm sure you can all imagine what the prefix mono means. Obviously it means one, but it means specifically one double bond. All right. So let's go ahead and draw one of these now. You're going to need to be, this year as a freshman, you're responsible for being able to identify these different monounsaturated and polyunsaturated fats. You will not have to draw them when you are a junior and a senior in IB biology. However, you will have to draw them. All right, so I went ahead and paused and drew this for you. Go ahead and draw this in your written notebooks, first of all, and then I'm going to dive into the different parts of it so you can pause and draw. As we take a look at this cis monounsaturated fat, okay, so we're going to, first of all, we're going to look here at the double bond, okay, and at the double bond, that's where we identify if it's cis. Well, first of all, every fatty acid is going to have a carboxyl group at the top, a carboxyl head, which is simply COOH. This is a molecule that you've seen throughout biochemistry so far. So we've got our carbon double bonded to our oxygen, single bonded to our hydroxyl or our OH group. Then we just have a long chain. We call this a hydrocarbon chain, right? A long hydrocarbon chain. And that is going to be how most all of these are going to look for us. All right. So then we find the double bond, and we're going to notice. So think back real quick. How many atoms can carbon bond to? That's one of the specific requirements, right, for something to be organic, is that it has to have carbon, and carbon is unique because it can bond to 4 other things. All right, so this carbon right here is going to have a double bond, so we're bonded to 1, 2, 3 things. Then we have our hydrogen up at the top. Those are the 4 things, the 4 atoms that carbon can bond to. That leaves an open area down here at the bottom, right? Same thing over here. So because this is a cis monounsaturated fat, the hydrogens are going to be on the same side, leaving this open area. So if you're looking at this on a multiple choice test, you're going to be looking for, if it's cis, you're going to be looking for something like this, where we have the hydrogens on the same side. This is going to cause this individual molecule to be able to bend. And that's actually what makes it healthy, is because it's able to bend. So when the blood flows through the coronary arteries towards the heart, these monounsaturated fats are able to pull the bad fats out because they're kind of sitting up. Think of it like if you were laying flat in a circular water slide, right? And the water is running over you. If you sit up, that's going to cause the water to push you through a little bit easier. It's very similar with blood flow in the coronary arteries. Our second type of unsaturated fat, also in the what we call the cis conformation, is going to be a cis polyunsaturated fat. And let's quickly review here, right? So cis means same side. Poly means that there's going to be multiple double bonds, and that it's an unsaturated fat. It's unsaturated because we have double bonds. So here you can see we have two double bonds, more than one. So there's not going to be like a cis diunsaturated. With unsaturated fats, it's either monounsaturated or polyunsaturated. Okay, so here we have our two areas where we're going to have a bend. All right, and this is going to actually be more healthy. So if you look at the nutrition label on a food item, so we can look at the nutrition label here on these healthy heart health nuts that Planters makes. Okay, so you can see here we've got a good amount of fat in these nuts, so you wouldn't want to eat them all throughout the day, but one serving of these a day would be considered to be very heart healthy. And let's go through this. So first of all, you can see there is 1.5 grams of saturated fat, but again, this is coming from plants, so this is not going to be the completely bad saturated fat that you might see like in a hamburger, for example. Okay, there's no trans fat, which is good, and we're going to look at trans fat next. So we've got cis unsaturated fats and we've got trans unsaturated fats. Okay, so the cis unsaturated fats are going to be the ones we're looking at first, and that falls under the polyunsaturated and the monounsaturated. Okay, so our polyunsaturated, that's going to be two double bonds at least, all right, perhaps more, and those are the healthiest possible fats that you can see when you're looking at your nutrition labels. The monounsaturated fats, those are healthy as well, so you can see why this is called the heart health mix. I try to have 1 bag of these a day, and it's a good way to try and continue to push the healthy fats through. And as we talked about, so what are some healthy foods that have a lot of this unsaturated fat? And you can see the contents here. We've got almonds, pistachios, pecans, walnuts. These are all things that are going to have good healthy fats. Again, this is going to be the cis unsaturated fat side. Now we're going to take a look at the trans unsaturated fats, the unhealthy ones. The bad side, this is what you're going to want to look for. You might want to pause and get this down. So for bad fats, we're going to call them unsaturated still, but these are trans unsaturated fats, right? We had our good side, all right, which were our regular mono and polyunsaturated fats, but on the cis side, so these are trans. So the foods we're going to see here are going to be things like hydrogenated vegetable oil, processed foods. We're looking at things like Twinkies, perhaps, things like that. French fries that are going to be extra hydrogenated with the oil that they're using in them. Chemically speaking, we're going to see that there are going to be double bonds. That's what makes them unsaturated. Okay, so we're looking at that there are double bonds. So that makes them unsaturated. When we say trans, it's trans because of the location of the double bonds, just like with the cis fats, right? So cis were on the same side, and then the trans are going to be transverse one another. So at those double bonds, the hydrogen bonds are going to be transverse one another. So let's take a look here at an example of this, or an example of what it would look like. Again, you need to be able to identify cis monounsaturated fats by looking at the picture, and then you're going to need to be able to identify trans mono and polyunsaturated fats by looking at a picture of them. So take a moment and go ahead and get this down. This is a drawing I just did of a trans unsaturated fat. This would be a trans mono unsaturated fat. You see I have my my carboxyl group at the top, just like I did with my previous example. The C OOH. I have that at the top. Okay, then I have my long hydrocarbon chain. I've got my double bond right here. I've got my hydrogens on the opposite side. They are transverse one another. All right, so go ahead and get this down, and then we'll discuss why these are so unhealthy. All right, so what I've drawn here, this would be representing a cross-section of a coronary artery. And each one of these black lines is going to represent a saturated fatty acid. We said that saturated fatty acids are the fatty acids that are found kind of in fried food, animal fat. The highest amount would be in beef, animal fat, okay? But they kind of lay flat. They don't have those double bonds that we saw in the unsaturated fats that help them to bend. And then here at the top you can see we have one that is bent. So this right here would be a cis monounsaturated fat. See how it bends up? So this, as the blood flows through, it's going to actually pick up this bent fatty acid over here and help to pull out some of these other fatty acids and help to reduce the cholesterol in the patient. So, for example, if you go to the doctor and your cholesterol is a little high, the first thing the doctor is going to do before prescribing some type of what we call a statin or a cholesterol lowering drug, they're going to ask you to improve your diet, improve exercise, and also to eat foods that are healthy and high in these unsaturated fats, these cis healthy unsaturated fats. As we continue with the bad fats then, so we've got our good fats were the unsaturated, the bad fats were the first part of the bad fats were the trans unsaturated fats, then we have our regular saturated fats. Food types for that, foods that are high in fried animal fat are going to fall into that. Dairy is going to be in this category, and animal fat. So go ahead and get those types down, and then we're going to take a look at why they are so bad from a chemical perspective. So from a chemical perspective, these saturated fats, we're going to see that they are completely saturated. Okay, that's why we call them saturated fats. So they are completely saturated. And what that simply means is that every carbon in the carbon chain is saturated with a hydrogen all the way around it. All right, in this hydrocarbon chain, that's what makes it saturated. We don't have any double bonds, so we don't have any open locations, right? If this was an unsaturated fat, this might be a double bond, which means that there would be no hydrogens down here. Okay, so that's what we mean when we say saturated fats. All right, now let's take a look at specifically how we would draw these. So we're still going to have our carboxyl group here at the top. We're going to have a hydrocarbon chain, and there's going to be no double bonds. All right, these are the types of fats that are going to help to make up the cell membrane, which is the phospholipid bilayer. And that's going to be the last part of today's lesson, is to take a look at the phospholipid bilayer and how it is composed and made up. Before we dive into the structure of the cell membrane, though, here's a good look of a little bit more of a shorthand version of these. So we've got, first of all, again, our saturated fatty acids. As you can see, there are no double bonds all through here, right? It's all single bonds. And then here's my carboxyl group here at the top. I've got my key down here for you of the different types of atoms. My unsaturated trans, the bad fats, you can see right here. This is my hydrocarbon chain, and right there I've got my double bond. I've got my carboxyl group here at the top, but at this double bond, notice that the hydrogens here in blue are on opposite sides. They are simply transverse one another. I've got my unsaturated cis fats here, and you can see here as we go through, I've got my hydrocarbon chain once again, guys, my carboxyl here, but here is my double bond, and you can see the hydrogens in blue are on the same side, or the cis side. So we've got the bent conformation. Again, these are going to be the healthy ones. All right, these are the unhealthy fats up here. All right, so now let's move into the plasma membrane of the cell. Okay, so taking a look at the phospholipid bilayer here. So we basically are zooming in. So if this is an animal cell right here, okay, in this picture, I'm zooming in on, let's say, this portion right here of the animal cell membrane, okay? And as you can see there, we are going to take a look at one individual phospholipid so far. So make sure that you get that terminology down, that it is called a phospholipid. So we've learned the structure of lipids, okay, and so we're zooming in on this. We've got this area right here is what we call the extracellular area, all right, which is outside of the cell. And then we have all of this fluid inside the cell, which is called the intracellular area. So the extracellular area is going to be called the interstitial fluid, and then the systolic fluid is going to be our intracellular area. All right, let's take a look here at the one individual phospholipid. So again, it's pieces making up larger parts. We've got the hydrophilic head. We need to know that hydrophilic means that something is attracted to water, okay, hydrophilic. And then hydrophobic, the hydrophobic tails are on the inside, which repel water. So if you have a phobia of spiders, that would be arachnophobia, right? So if you're afraid of it, so something that's hydrophobic is going to repel water. These also, we can take a look and identify which one is polar and which one is nonpolar. So take a moment to brainstorm which one of these is going to be polar, the hydrophilic head or the hydrophobic tail. So we should have that the head of this is going to be polar, hence why it is attracted to water, right? And then our hydrophobic tails are going to be considered non-polar. All right. So those are the individual units that are making up the cell membrane. All right. So the first thing here, I don't want you to draw this big fancy picture up here. We're just going to use that as a model, okay? There's a couple things, though, that I would like to make sure that you get down. I'm going to highlight those for you. So first of all, again, you should have already drawn this, that we have our polar hydrophilic phosphate head, our fatty acid tails. Those fatty acids are just like the fatty acids that we just learned about in the beginning of this lesson. This is what we do need to get down, the components of the plasma membrane. The first component is a phospholipid. All right, phospholipids, they form from a bilayer with phosphate heads, and then the fatty acid tails that are turned inward. Again, they're turned inward because they're hydrophobic. They're afraid of the water that is going to be surrounding the cell. Remember, most living organisms, well, I guess all living organisms, are primarily composed of water. That's why we spent so much time learning about the properties of water. Next, we're going to have our cholesterol, which is found in animal cell membranes. That's going to be embedded into the cell membrane to help it with its structure. And then we're going to have proteins that are going to help to move things in and out, like our channel protein here, which we would see with facilitated diffusion, or a carrier protein, which would be active transport, pushing things against the concentration gradient. So a lot of you have already seen examples of this. Freshman bio, you guys just need to be able to identify the parts of the cell membrane. Junior and senior bio, you might need to actually draw this part of the plasma membrane. So keep that in mind depending on which class you're in. And lastly, we're going to take a look at the fluid mosaic model, which is a kind of a three-dimensional model of the cell membrane. Think of a mosaic painting, for example, something that has individual tiles that kind of are pieces to the large painting. That's where this comes from overall. So it's just showing you that we put all of these pieces and components together, and that puts together the fluid mosaic model. The next part of this lesson today is going to be to use an online resource, which will be from Khan Academy. And there's a very good 8-minute video on Khan Academy about the fluid mosaic model, and it will also help to reinforce a lot of the key principles that I've just taught you on the cell membrane. So that's the second part of today's lesson. Hope you took good notes and got a lot out of this. And next you're going to flip over to the Khan Academy lesson.